Suppose you had a paper cut on your finger, and although the cut may have bled and stung for a while, after a few days it will have disappeared and healed. So consider the following three questions. How do you think the body repairs such an injury? How long do you think the repair process will continue? And what do you think causes the cells to stop the repair process? In thinking about these questions, scientists developed two experiments. The first was to apply cultures of cells on nutrient medium in petri dish, and the cells continued to divide until they filled the dish. Then an injury was inflicted by scraping away some cells down the middle. You might consider then that the cells along the margin of the injury began to divide until the cell petri dish was filled again. Based on the experimental results, you might suggest that the controls for the cell cycle were external in origin, meaning that cells, when they come in contact with each other, stop dividing. This is called contact inhibition. But a second experiment, one that involved removing cytoplasm from a cell in metaphase, really changes this perspective as well, because that cytoplasm was then injected into a different cell that was in interphase, and with that injection, the cell began to initiate a new mitotic process. So in this case, we see that the cell cycle is controlled by internal controls, or internal regulation, and this is by protein compounds referred to as cyclins, and they control checkpoints along the cell cycle. There are three checkpoints we're most interested in. The first is called the G1 checkpoint, and it's concerned with evaluating the two questions: is the cell big enough, and is the environment suitable, or is there space available? Think of this as a lot like a traffic control signal. Cells that can answer these questions in the affirmative move on to the next stage. If they cannot, then they stop and go back to G1, where they may continue to grow or continue to perform their cell activities. Some cells remain in the G0 stage for their entire existence. The second checkpoint is called the G2 checkpoint, where DNA is concerned with. The next checkpoint is called the G2 checkpoint, which is primarily concerned with DNA being copied correctly. The third checkpoint is called the metaphase checkpoint, which is concerned with the spindle fiber being attached to the centromeres. Unlike the G1 checkpoint, where a cell that fails the checkpoint would simply go back to a G1, the G2 and metaphase checkpoint initiate a process called apoptosis, where programmed cell death eliminates the cell from any further problems. If the G2 and the metaphase checkpoints aren't satisfactorily completed, the cell is just destroyed. Examine the following data. The lifespan of various human cells are displayed on the data table, along with their division process and the type of the cell. Answer the following four questions, and when you're complete, answer then the reflection question. Be sure to explain your answer.